Hi everyone, this is Dr. Santosh. Today we will talk about Chargaff Rule and nucleotide base pairing. So before we talk about that, let's talk about the interesting history behind the Chargaff Rule and this double-stranded DNA. So what happened is the DNA in early 1920s, it was not considered as genetic material. People after looking at DNA, they did not think that DNA could be the genetic material. But Griffith and every McCarthy and Hasic's experiments, what they concluded that DNA is the genetic material. They established DNA as genetic material. So after it has been established, there was a rush in discovering the structure of DNA. So in meantime, there was a scientist called Chargaff. You can see he was an Austrian scientist working in USA. He's a biochemist. Now, what he did is he analyzed the DNA of many species, and what he found is in his DNA, what he found is many of the species found from bacteria till eukaryotes in human also. They find one common thing, that they found that percentage of adenine and percentage of thymine are same. The same thing with guanine and cytosine. And then mark that this adenine and guanine, when they add up this adenine and guanine together, it was equal to cytosine and thymine. He proposed his finding: percentage of adenine is equal to percentage of thymine, and percentage of guanine is equal to percentage of cytosine. First, and then adenine plus guanine, that is purine, is equal to pyrimidine. Now he did this work in 1940s, late 1940s, and he published paper in 1950s. What at that time there was a rush for discovery of double-stranded helical structure. The Watson, Crick, as you know, Watson, Crick, and Wilkins, they got the Nobel Prize for double-stranded helical in 1960s. But they discovered double-stranded helical structure. They actually did not do that big lab work. The lab work, they used the foundation of lab work from different scientists did before. One of them are Chargaff. He used the work of Chargaff, and not only Chargaff, he also used the work, mainly the work of Rosalind Franklin. She was a very eminent personality at that time. She was doing X-ray crystallography of DNA. The third scientist, that Wilkins, he used to take all the data from Rosalind Franklin and give it to Watson and Crick, and Watson and Crick used those data to figure out the structure of DNA. And then later, this Erwin Chargaff also made Watson and Crick in 1952 and explained his finding. And when he explained finding, his finding was one of the cornerstone for the discovery of double-stranded helical structure. Now, they put all those data together and they came up with the discovery of double-stranded helical structure. Though Chargaff gave this principle that adenine is equal to thymine, guanine is equal to cytosine, adenine plus guanine, that is purine, is equal to pyrimidine, but he did not make a link between this adenine and thymine. He could not make a link with the structure of DNA, that these two bases, how they form hydrogen bond. The Watson and Crick, what they did, they tried to find out the link, and they gave this hydrogen bond. They found that, especially Watson, what he found is, in the course of making the model of double-stranded helical structure, what he did, he put the base together, and then he found that there are possibility of 3 hydrogen bond formation in guanine and cytosine, and there is possibility of formation of 2 hydrogen bond between adenine and thymine. And that is how the hydrogen bonding came into existence. These two bases pair each other through hydrogen bond was given by Watson and Crick. And this is how, based on their finding, double-stranded helical structure was formulated. And after that, what happened is this: after they got Nobel Prize, this Erwin Chargaff also wrote the letter to Nobel Prize committee that he should also be given credit for that, but he did not get any credit for Nobel Prize. But we should admire his work. His work actually laid the foundation for discovery of double-stranded helical structure, and that opened the discovery of double-stranded helical structure opened all Pandora box for the molecular biology. Now, the molecular biology today we see here is because of the discovery of double-stranded helical. It is the first very important cornerstone that Watson and Crick put based on the data of Erwin Chargaff, Rosalind Franklin, and other scientists. So you can see here this two base adenine and thymine and cytosine and guanine, they form base pair, and the base pairing is because of hydrogen bonding, and hydrogen bonding is because of side chains of their adenine. So you can see in adenine adenine has that is the importance of side chain. You need to know the importance of side. You need to know the side chain. So that will help you understand how it forms the base pair with thymine. Adenine forms base pair with the thymine, and guanine forms base pair with the cytosine. You can see in adenine forms NH group. H form the base pairing hydrogen bond. It is a hydrogen bond is a non-covalent bond. It is just an attraction. So we see, and where you can see one hydrogen will be there. Okay, this is partially positive hydrogen will be attracted towards partially negative oxygen here. Now partially negative nitrogen will attract this partially positive hydrogen here. Now, this adenine and thymine, this is how it will form two hydrogen bond. And this here you can see in cytosine and guanine, it will form three hydrogen bond here. The guanine, it has got oxo group here, and at here you can see in second carbon it has got this NH2 group and will form this hydrogen bond with this oxo group. This is how the cytosine and adenine form three hydrogen bonds. The significance of this hydrogen bond is immense. The first very important thing is this hydrogen bond is very important for the stability of DNA. Okay, stability of DNA. Now, and base pairing is very important during DNA synthesis, because this base pairing helps the selection of a particular type of nucleotide. Okay, so this is called kinetic selectivity. So this kinetic selectivity is helped by this base pair. DNA and RNA polymerase cannot select this, so base pairing helps that. Okay, template strand is there, and the base pairing with the template strand will choose a proper type of nucleotide sequence for DNA and RNA synthesis. And base pairing is a very important tool that we use. Understanding of that base pairing we utilize for our polymerase chain reaction. So you can see in polymerase chain reaction we use this formula. Okay, so we find out the melting temperature of the DNA probe through this formula, and this G and C base pairing, because there is three hydrogen bonds, so G and C base pairing decides the melting temperature. It is a determining factor. So we need GC bonding, but we do not need too much of GC bonding, and we decide this. We use this principle. We use this base pairing understanding, knowledge of this base pairing to design probe. So now base pairing is also utilized. This base pairing is also utilized to open the DNA double helix. Okay, during replication and also during transcription at promoter region. So you will find in promoter region and the origin of replication there will be A and T region. The A and T base will be more, because those places are very easy for the enzymes to break and starts replication and transcription. One thing, the Chargaff rule is not applicable in single-stranded DNA, which is present in DNA virus. There are single-stranded DNA virus which is called parvovirine. So thank you for today.